Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Bacillus anthracis derives its name from bacillus, meaning little rod, and anthracis, which refers to coal. Thus, Bacillus anthracis is a rod-shaped bacterium responsible for the disease anthrax, known for causing distinctive black skin lesions. Historically, Bacillus anthracis, often abbreviated as B. anthracis, has been responsible for several epidemics in Europe and has been utilized as a biological weapon. Bacillus anthracis features a thick peptidoglycan cell wall that absorbs purple dye during gram staining, classifying it as gram-positive. This bacterium is non-motile and a facultative anaerobe, allowing it to thrive with or without oxygen. Additionally, B. anthracis is non-beta hemolytic. When grown on blood agar, its colonies do not induce beta hemolysis, which is the breakdown of red blood cells that would cause the blood agar to change color from red to transparent yellow. Bacillus anthracis is a spore-forming bacterium capable of endosporulation when faced with harsh environmental conditions, such as extreme temperatures, dryness, or harmful radiation. During endosporulation, the bacterium begins by replicating its DNA, then forms a protective wall inside the cell, creating a larger structure called the mother cell and a smaller portion known as the forespore. The plasma membrane then surrounds the forespore and pinches it off, resulting in the formation of a separate forespore. The forespore is subsequently engulfed by the mother cell, leading to a cell-within-a-cell arrangement. Within the deteriorating mother cell, the forespore dehydrates and accumulates calcium while being encased in a robust cortex. This process endows the endospore with exceptional resistance to heat, harsh chemicals, digestive enzymes, and antibiotics due to the presence of dipicolinic acid in the core. Eventually, the mother cell dies, releasing the endospore into the environment. Remarkably, an endospore can endure for over 1,000 years, remaining dormant until conditions become favorable again. When conditions improve, the endospore can germinate into the active bacterial form, which can then grow, divide, and cause anthrax infection. In humans, anthrax can affect the skin, lungs, or gastrointestinal tract. Bacillus anthracis initially enters the body as endospores, which are engulfed by resident macrophages. For instance, when the bacteria reach the alveoli, tiny air sacs in the lungs where gas exchange occurs, they are consumed by lung macrophages and then transported through lymphatic vessels to the adjacent mediastinal lymph nodes. A similar process happens in the skin and gastrointestinal tract. Within the macrophage, the spore germinates, transforming into the active bacterial form. The bacteria then exit the macrophage through cytolysis, which involves the rupture of the cell membrane, causing the cell to burst and release the bacteria into the surrounding tissues. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. In terms of pathogenesis, the active form of Bacillus anthracis first produces a poly-D-glutamic acid capsule with anti-phagocytic properties, preventing the bacteria from being ingested by macrophages once they are released from the initial macrophage. The bacterium then secretes anthrax toxin, which consists of 3 proteins: protective antigen, lethal factor, and edema factor. Protective antigen is released from the bacteria and binds to immune cells via cell surface proteins known as either ANTXR1 or tumor endothelial marker 8. The name protective antigen reflects its role in immunity. Individuals with antibodies against this protein are considered immune to B. anthracis. Once protective antigen binds to the immune cell, it forms a channel in the cell membrane, allowing the lethal factor and edema factor proteins to enter the intracellular space. Lethal factor is a zinc metalloprotease, a type of protein that uses zinc as a cofactor to cleave mitogen-activated protein kinases 1 and 2. These enzymes play a crucial role in helping cells produce NADPH, an important cofactor in various biochemical pathways that facilitate energy generation in the form of ATP. When lethal factor cleaves these enzymes, the cells are unable to produce ATP, ultimately leading to cell death through apoptosis or programmed cell death. Edema factor, named for the edema it causes around the black 𝐂𝐴𝐧𝐭𝐞𝐳, contains ADP ribosyl-transferase. This enzyme converts adenosine triphosphate, ATP, into cyclic adenosine monophosphate, 𝐂𝐴MP. 𝐂𝐴MP activates the protein kinase A signaling pathway, influencing various cellular functions by either activating or deactivating them. A key effect of this pathway activation is the disruption of the cell's ability to secrete cytokines, which are signaling molecules that recruit white blood cells to combat the infection. As a result, the bacteria continue to proliferate and destroy immune cells in the lymph nodes, impairing the immune response. This leads to surrounding tissue necrosis and eventual bleeding. With access to the bloodstream, the bacteria can spread to multiple organs, causing extensive necrosis. This progression results in multi-organ failure, septic shock, and ultimately, death. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Clinically, anthrax manifests in 3 forms. The most common form is cutaneous anthrax, which occurs when endospores come into contact with an open wound in the skin, often through contact with animal hair or wool. In this form, the endospores germinate locally, producing additional spores and active bacteria. This leads to the development of painless small red bumps or papules that eventually evolve into a large vesicle or fluid-filled sac, which then ruptures. Following rupture, the surrounding tissue undergoes necrosis, resulting in a characteristic painless black lesion known as an 𝐓𝐚𝐿. Gastrointestinal anthrax arises when an individual consumes undercooked meat contaminated with anthrax spores. These spores can infect any part of the gastrointestinal tract, from the mouth to the rectum. Similar to the skin form, the spores can lead to the formation of black eschers in the GI tract. Symptoms typically include abdominal pain, nausea, and vomiting, often with blood, along with diarrhea or bloody diarrhea. Respiratory anthrax, also referred to as wool sorter's disease, occurs when anthrax spores are inhaled. In this form, black eschars do not appear in the lungs, but rather in the surrounding mediastinal lymph nodes. This can cause pulmonary hemorrhage, severe necrosis of the mediastinal lymph nodes, and bacteremia, where bacteria enter the bloodstream. Symptoms include coughing up bright red blood, high fevers, low blood pressure, and a rapid heart rate. Unfortunately, respiratory anthrax is associated with the highest mortality rate among the forms of anthrax. Diagnosing anthrax can be challenging due to its rarity and the fact that many hospitals may lack the necessary diagnostic tools. The diagnostic methods used for anthrax include a polymerase chain reaction, PCR, assay, which detects B. anthracis DNA directly, and immunohistochemical staining of tissues, which identifies B. anthracis antigens in tissue samples. Additionally, an anti-protective antigen immunoglobulin assay detects antibodies against the protective antigen of the bacterium. Other diagnostic indicators may include a widened mediastinum observed on a chest X-ray, which suggests progressive necrosis of the mediastinal lymph nodes. The treatment of anthrax involves a combination of intravenous antimicrobial therapy, anthrax antitoxin, and supportive care. The primary antimicrobial agents include ciprofloxacin, used in conjunction with either clindamycin or linezolid. Additionally, monoclonal antibodies such as raxibacumab and obiltoxaximab, which specifically target and inhibit the protective antigen, should be administered to individuals confirmed to have fulminant systemic anthrax. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Bacillus Anthracis Summary. Bacillus anthracis is a rod-shaped, gram-positive, spore-forming bacterium that causes the disease anthrax. The spore form enables B. anthracis to endure harsh environmental conditions and is also the infectious form of the bacterium. B. anthracis can lead to respiratory, gastrointestinal, or cutaneous anthrax, utilizing virulence factors such as protective antigen, lethal factor, and edema factor. All three forms have the potential to progress to fulminant disseminated anthrax. Diagnosis is confirmed through polymerase chain reaction, PCR, immunohistochemical staining, or anti-protective antigen immunoglobulin assays. Treatment includes intravenous antimicrobial combination therapy, anthrax antitoxin, and supportive care. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you.